EPSCs provide a platform to study early embryo cells in detail at a molecular level to understand development not only in mouse, but ultimately in future in humans. This new method of producing stem cells could be enormously helpful for studying development more efficiently, generating functional human cells, and researching treatments for pregnancy problems such as pre-eclampsia and miscarriages. And as a fertilized egg develops into a blastocyst, it produces cells that will form the embryo, where ES cells come from, and two other types of cell that will develop into the placenta or the yolk sac. It is possible to establish 3 different types of stem cells, including ES cells from these 3 cell types in blastocysts. EPSCs are the first stem cell that are able to produce all these 3 types of blastocyst stem cells, which give them much greater potential for development. And to discover new stem cells for use in research in regenerative medicine, the researchers created a way of culturing cells from the earliest stage of development. When the fertilized egg has only divided into 4 or 8 cells that are still considered to retain some tautopotency, the ability to produce all cell types. Their hypothesis was that these cells should be less programmed than ES cells, which are taken from the around 100 cell stage of development. Called a blastocyst, they grew these early cells in a special growth condition that inhibited key development signals and pathways. And published today in Nature 11th October, the methods used could also help produce similar stem cells lines from human and other mammalian species, including those such as pigs or cows, where embryonic stem cells lines are still not available. The researchers also believe that their study could have implications for human regenerative medicine and for understanding miscarriage and developmental disorders. Stem cells have the ability to develop into other cell types, and existing cells lines are already extremely useful for research into development, disease, and treatments. However, the 2 currently available types of stem cell lines, embryonic stem cell ES and induced pluripotent stem cells IPS, have certain implementations. It is not currently possible for them to form every type of cell, since they are already excluded from developing certain cell lineages. And today's Dr. Will, CBMD PhD case, Sunday, January 21st, 2018, 10:24 a.m. Eastern Standard Time. Science News. New type of stem cell line produced offer expanded potential for research and treatments. Researchers have created expanded potential stem cells which could shed light on miscarriage and development disorders. Date October 11, 2017. Wellcome Trust, Sanger Institute. Researchers have created expanded potential stem cells, EPSCs, in mice for the first time that have greater potential for development than current stem cell lines. These stem cells have the features of the very first cells in the developing embryo and can develop into any type of cell. And plant and animal breeders are also employing the techniques of molecular genetics to introduce genes from other species into crop, plant, and livestock. These mapping projects have been carried on relentlessly, and for one species, rice, have culminated in the complete sequencing of the genome. Other crop and livestock genome sequencing projects are still in progress. All sorts of potentially useful genes are being identified and studied in these projects. Beginning in the 1980s, classical approaches to crop and livestock improvement were supplemented and in some cases supplanted by approaches from molecular genetics. Detailed genetic maps of the chromosomes of several species were constructed to pinpoint genes of agricultural significance. By locating genes for traits such as grain yield or disease resistance, breeders could now design schemes to incorporate particular allies into agricultural varieties. Selective breeding programs now informed by genetic theory continue to play important roles in agriculture. High-yielding varieties of wheat, corn, rice, and many other plants have been developed by breeders to feed a growing human population. Selective breeding techniques have also been applied to animals such as beef and dairy cattle, swine, and sheep, and to historical plants such as shade trees, turf grass, and garden flowers. For example, cattle were changed in appearance and behavior, and corn, which is descendant from wild grass called teosinte, was changed so much that it could no longer grow without human cultivation. By the time the first civilizations appeared, humans had already learned to cultivate crop plants and rear livestock. They had also learned to improve their crops and livestock by selective breeding. This pre-Mendelian application of genetic principles had telling effects. over thousands of generations, domesticated plant and animal species came to be quite different from their wild ancestors. There are also evidence in the myriad ways in which genetics is applied in agriculture, medicine, and many other human endeavors all over the world. And genetics in the world. Applications of genetics of human endeavors. Genetics relevant in many venues outside the research laboratory. Genetics began in European monostatic enclosure today in its worldwide enterprise. The significance and international scope of genetics are evident in today's scientific journals, which showcase the work of geneticists from many different countries. Key points. In classical genetic analysis, genes are studied by following the inheritance of traits and crosses between different strains of an organism. In molecular genetic analysis, genes are studied by isolating, sequencing, and manipulating DNA, and by examining the products of gene expression. In population genetic analysis, genes are studied by assessing the variability among individuals in a group of organisms. And key points. In classical genetic analysis, genes are studied by following the inheritance of traits and crosses between different strains of an organism. In molecular genetic analysis, genes are studied by isolating, sequencing, and manipulating DNA, and by examining the products of gene expression. In population genetic analysis, genes are studied by assessing the variability among individuals in a group of organisms. The assessment of genetic variability in a population is therefore a foundation for the study of biological evolution. It is also useful in the effort to understand the inheritance of complex traits such as body size or disease susceptibility. Often complex traits are of considerable interest because they have an agricultural or medical significance. Geneticists seek to document this variability and to understand its significance. Their most basic approach is to determine the frequencies of specific allies in population and then to ascertain if these frequencies change over time. If they do, the population is evolving. Genetics can also be studied at the level of an entire population of organisms. Individuals within a population may carry different allies of a gene. Perhaps they carry different allies of many genes. These differences make individuals genetically distinct, possibly even unique. In other words, the members of a population vary in their genetic makeup. They have even learned how to transfer genes from one organism to another. These recombinant DNA molecules can be replicated in a bacterial cell or even in test tubes that have been supplied with appropriate enzymes. Milligram quantities of a particular gene can be generated in the laboratory in the afternoon. In short, geneticists have learned how to manipulate genes more or less at will. This artful manipulation have allowed researchers to study genetic phenomena in great detail. But the molecular approach to genetic analysis is much more than the study of DNA sequences. The geneticists have learned to cut DNA molecules at specific sites. Whole genes or pieces of genes can be excised from one DNA molecule and inserted into another DNA molecule. The gene's internal components, coding sequences, regulatory sequences, and non-coding sequences can be identified. The nature of the polypeptide encoded by the gene can be predicted. And molecular genetics. With the discovery of the structure of DNA, genetics entered a new phase. The replication, expression, mutation of genes can now be studied at the molecular level. This approach to genetic analysis was raised to a new level when it became possible to sequence DNA molecules easily. Molecular genetics analysis is rooted in the study of DNA sequences. Knowledge of DNA sequence and comparisons to other DNA sequences. Because these studies emphasize the transmission of genes and chromosomes from one generation to the next, they are often referred to as exercises in transmission genetics. However, classical genetics is not limited to the analysis of gene and chromosome transmission. It also studies the nature of the genetic material. However, it controls the traits and how it mutates. And the classical approach to the study of genes can also be coordinated with studies on the structure and behavior of chromosomes, which are the cellular entities that contain the genes. By analyzing patterns of inheritance, geneticists can localize genes to specific chromosomes. More detailed analysis allows them to localize genes to specific positions within chromosomes, a practice called chromosome mapping. The trait differences are due to the alternate forms of genes. Sometimes more than one gene influences a trait, and sometimes environmental conditions, for example temperature and nutrition, exert an effect. These complications can make the analysis of inheritance difficult. Classical genetics. The period prior to the discovery of the structure of DNA is often spoken of as the era of classical genetics. During this time, geneticists pursued their science by analyzing the outcomes of crosses between different strains of organisms, much as Mendel had done in his work with peas. In this type of analysis, genes are identified by studying the inheritance of trait differences, tall pea plants versus short pea plants, for example, in the offspring of crosses. And all these levels of genetic analysis are routinely used in research today, although we shall encounter them in many different places in this book. We provide brief descriptions of them in here. Still, another type of genetic analysis imitates Darwin and Wallace by focusing on entire populations of organisms. Genetic analysis is practiced at different levels. The oldest type of genetic analysis follows a Mendel's footstep by focusing on how traits are inherited when different strains of organisms are hybridized. Another type of genetic analysis follows in the footsteps of Watson and Crick and the army of people who have worked on the various genome projects by focusing on the molecular makeup of the genetic material. And levels of genetic analysis. Geneticists approach their science through different points of view, from that of a gene, a DNA molecule, or a population of organisms. Key points. Evolution depends on the occurrence, transmission, and spread of mutant genes in groups of organisms. DNA sequence data provide a way of studying the historical process of evolution. Today, the construction of phylogenetic trees is an important part of the study of evolution. Biologists use the burgeoning DNA sequence data from the Genome Project and other research ventures, such as the United States National Science Foundation, Tree of Life, program in combination with anatomical data collected from living and fossilized organisms to discern the evolutionary relationships among species. Organisms with very similar DNA sequences are descended from the recent common ancestor. Organisms with less similar DNA sequences are descended from a more remote common ancestor. Using this logic, researchers can establish the historical relationship among organisms. With DNA sequencing, we can see similarities and differences in genetic material of diverse organisms. On the assumption that sequences of nucleotides in the DNA are the result of historical processes, it is possible to interpret these similarities and differences in a temporal framework. Research on biological evolution was stimulated when Mendel's discoveries came to light at the beginning of the 20th century, and it took a new turn when DNA sequencing techniques emerged at the century's end. The idea of Darwin and Wallace revolutionized scientific thought. They introduced a historical perspective into biology and gave credence to the concept that all living things are related to by virtue of descent from a common ancestor. However, when these ideas were proposed, Mendel's work on heredity was still in progress and the science of genetics had not yet been born. In the middle of the 19th century, Charles Darwin and Alfred Wallace, both contemporaries of Mendel, proposed that this variation makes it possible for species to change, that is, to evolve over time. And gradually, over long stretches of time, mutations accumulate in the DNA, and we see their effects and differences among organisms. Mendel's strains of peas carry different mutant genes, and so do people from different ethnic groups. In almost any species, at least some of the observable variations has an underlying genetic basis. Answers to these questions must take into account the more or less faithful replication of DNA and its transmission from generation to generation, however mutations occur, and the resulting mutant genes are copied and passed on to subsequent generations. And what aspects of the genetic material make us human? How has this material come down to us from our ancestors? How is it changed over time? Is our genetic material related to that of other species? Is our history part of the larger history of life? Answers to these questions must take into account the more or less faithful replication of DNA and its transmission from generation to generation. And at some point, however, even the archaeological evidence for human activity vanishes, and we are left with the questions: who are we? Where do we come from? These questions about the identity and origin of human beings force us to think about our genetic heritage. Recorded human history spans a few thousand years. Artifacts, cave paintings, arrowheads, stone and bone tools, and other materials give us a glimpse of human life deeper in the past. For most genes, RNA synthesis transcription generates a molecule, the RNA transcript, that becomes a messenger RNA, mRNA. Coded information in an mRNA is translated into a sequence of amino acids and polypeptide. Mutations can alter the DNA sequence of a gene. The genetic variability created by mutation is the basis for biological evolution. And when DNA replicates, each strand of a duplex molecule serves as a template for the synthesis of a complementary strand. When genetic information is expressed, one strand of a gene's DNA duplex is used as a template for the synthesis of a complementary strand of RNA. And the process of mutation has another aspect. It introduces variability into the genetic material of organisms. Over time, the mutant genes created by mutation may spread through the population, much like the mutant B globin gene has spread in some human populations. These changes in the genetic composition of a population are the basis of biological evolution. And people who carry only the mutant version of the B globin gene have sickle-shaped red blood cells, whereas people who carry only the non-mutant version of this gene have disc-shaped red blood cells. The sickle-shaped cells do not transport oxygen efficiently through the body. Consequently, people with sickle-shaped red blood cells develop a serious disease, so serious in fact that they eventually die from it. This disease, called sickle cell anemia, is therefore traceable to a mutation in the B globin gene. We shall investigate the nature of the causes of mutations like this one in Chapter 13. This mutation, now widespread in some human populations, alters the 6 codon so that it specifies the incorporation of valine into the B globin polypeptide. This seemingly insignificant change has a deleterious effect on the structure of the cells that make and store hemoglobin, the red blood cells. And the 146 amino acids in B globin correspond to 146 codons in the B globin gene. The 6 of these codons specifies the incorporation of glutamic acid into the polypeptide countless generations ago in the germ line of some nameless individual. The middle nucleotide pair in this codon was changed from AT to TA, and the resulting mutation was passed on to the individual's descendants. And often mutant genes cause different traits in organisms. Figure 1.9, for example, one of the genes in the human genome encodes the polypeptide known as B globin. This polypeptide, 146 amino acids long, is a constituent of hemoglobin, the protein that transports oxygen in the blood. And DNA molecules are also sometimes damaged by electromagnetic radiation or by chemicals. Although the damage induced by these agents may be repaired, the repair process often leaves scars. Stretches of nucleotides may be deleted and duplicated, or they may be arranged within the overall structure of the DNA molecule. We call all these types of changes mutations. Genes that are altered by the occurrence of mutations are called mutant genes. And mutation-changing genetic information. DNA replication is an extraordinarily accurate process, but it is not perfect. At a low but measurable frequency, nucleotides are incorporated incorrectly into growing DNA chains. Such changes have the potential to alter or disrupt the information encoded in genes. In several chapters we shall see circumstances in which the first part of this progression is reversed. That is, RNA is used as a template for the synthesis of DNA. This process, called reverse transcription, plays an important role in the activities of certain types of viruses, including the virus that causes acquired immune deficiency syndrome, or AIDS. It also profoundly affects the content and structure of the genome of many organisms, including the human genome. And from all these considerations, it is clear that information flows from genes, which are composed of DNA, to polypeptides, which are composed of amino acids, through an interaction intermediate, which is composed of RNA. Thus, in the broad sense, the flow of information is DNA-RNA polypeptide, a progression often spoken of as the central dogma of molecular biology. The study of the proteins in cells, their composition, the sequence of amino acids, and their constituent polypeptides, the interactions among these polypeptides and among different proteins, and of course the functions of these complex molecules, is called proteomics. Like genomics, proteomics has been made possible by advances in the technologies used to study genes and gene products, and by the development of computer programs to search databases and analyze amino acid sequences. Some polypeptides are altered by the removal of the first amino acid, which is usually methionine, in the sequence. We refer to the collection of all the different proteins in an organism as its proteome. Humans with between 20,000 and 25,000 genes may have hundreds of thousands of different proteins in their proteome. One reason for the large size of the human proteome is that a particular gene may encode several different but related polypeptides, and these polypeptides may combine in complex ways to produce different proteins. Another reason is that proteins may be produced by combining polypeptides encoded by different genes. If the number of genes in the human genome is large, the number of proteins in the human proteome is truly enormous. The second stage in the expression of the gene's information is called translation. At this stage, the gene's mRNA acts as a template for the synthesis of a polypeptide. Each of the gene's codons now present in the sequence of the mRNA specifics the incorporation of a particular amino acid into the polypeptide chain. One amino acid is added at a time. Thus, the polypeptide is synthesized stepwise by reading the codons from one end of the mRNA to the other. When the polypeptide is finished, it dissociates from the mRNA and carries out its role in the cell. The finished molecule, called the messenger RNA or simply mRNA, contains all the information needed for the synthesis of a polypeptide. The expression of genetic information to form polypeptide is a two-stage process. First, the information contained in a gene's DNA is copied into a molecule of RNA. The RNA is assembled in a stepwise fashion along one of the strands of the DNA duplex. During the assembly process, A in the RNA pair with T in the DNA, G in the RNA pairs with C in the DNA, C in the RNA pairs with G in the DNA, and U in the RNA pairs with A in the DNA. Thus, the nucleotide sequence of the RNA is determined by the nucleotide sequence of a strand of DNA in the gene. The process that produces this RNA molecule is called transcription, and the RNA itself is called transcript. The RNA transcript eventually separates from its DNA template and in some organisms is altered by the addition, deletion, or modification of nucleotides. Thus, the information encoded within a gene is used to direct the synthesis of a polypeptide, which is often referred to as the gene's product. Sometimes, depending on how the coding information is utilized, a gene may encode several polypeptides. However, these polypeptides are usually all related by sharing some common sequence of amino acids. The sequence of amino acids in polypeptide is specified by a sequence of elementary coding units within a gene. These elementary coding units, called codons, are triplets of adjacent nucleotides. A typical gene may contain hundreds or even thousands of codons. Each codon specifies the incorporation of amino acid into a polypeptide. Each polypeptide has a characteristic of sequences of amino acids. Some polypeptides are short, just a few amino acids long, whereas others are enormous, thousands of amino acids long. How is the information within individual genes organized and expressed? This question is central in genetics, and we will turn our attention to it in Chapter 11, 12, and 14. Here suffice it to say that most genes contain the instructions for the synthesis of proteins. Each protein consists of one or more polypeptides, which are chains of amino acids. The 20 different kinds of amino acids that occur naturally can be combined in a myriad ways to form peptides. Most of the DNA in M genitalium comprises genes, whereas most of the DNA in human beings does not. That is, most of the human DNA is non-genetic. We shall investigate the genetic and non-genetic composition of genomes in many places in this book, especially in Chapter 16. Each gene is stretched of nucleotide pairs along the lengths of DNA molecule. A particular DNA molecule may contain thousands of different genes. In an M genitalium cell, all the genes are situated on one DNA molecule. The single chromosome of this organism. In a human sperm cell, the genes are situated on 23 different DNA molecules corresponding to the 23 chromosomes in the cell. In these and all other genomes, the information contained within the DNA is organized into the units we call genes. An M genitalium has 482 genes, whereas a human sperm has between 20,000 and 25,000. Each gene is a stretch of nucleotide pairs along the length of a DNA molecule. This information is encoded in sequences of nucleotides within the DNA molecules of the genome. Among cellular organisms, the smallest known genome is that of Mycoplasma genitalium, 5,080,070 nucleotide pairs. By contrast, the human genome consists of 3.2 billion nucleotide pairs. Gene expression using genetic information. DNA molecules contain information to direct the activities of cells and to guide the development, functioning, and behavior of the organisms that comprise these cells. At the end of the replication process, each template strand is paired with a newly synthesized partner strand. Thus, two identical DNA duplexes are created from one original duplex. The process of DNA replication does not occur spontaneously. Like most biochemical processes, it is catalyzed by enzymes. We shall explore the details of DNA replication, including the roles played by different enzymes. This technique incorporation conforms to the base pairing rules. Thus, the sequence of nucleotides in the strand being synthesized is dictated by the sequence of nucleotides in the template strand. This incorporation conforms to the base pairing rules. Thus, the sequence of nucleotides in the strand being synthesized is dictated by the sequence of nucleotides in the template strand. The process of DNA replication is based on the complementary nature of the strands that make up double DNA molecules. These strands are held together by hydrogen bonds between specific base pairs, a paired with T and G paired with C. When these bonds are broken, the separated strands can serve as templates for synthesis of new partner strands. The new strands are assembled by the stepwise incorporation of nucleotides opposite to nucleotides in template strands. DNA replication propagating genetic information. The genetic material of an organism is transmitted from a mother cell to its daughters during the cell division. It is also transmitted from parents to their offspring during reproduction. The faithful transmission of genetic material from one cell or organism to another is based on the ability to double-stranded DNA molecules to be replicated. DNA replication is extraordinarily exact. Molecules consisting of hundreds of millions of nucleotide pairs are duplicated with few, if any, mistakes. DNA is a genetic material. The central dogma in biology is that information flows from DNA to RNA to protein. In all cellular organisms, the genetic material is DNA. This material must be able to replicate so that copies can be transmitted from cell to cell and from parents to offspring. It must contain information to direct cellular activities and to guide the development, functioning, and behavior of organisms, and it must be able to change so that, over time, groups of organisms can adapt to different circumstances. DNA is a hereditary material in all life forms except some types of viruses in which RNA is a hereditary material. The Human Genome Project determined the sequence of the nucleotide in the DNA of the human genome. Sequencing the DNA of a genome provides the data to identify and catalog all the genes of an organism. Gregor Mundell postulated the existence of genes to explain how traits are inherited. Allies, the alternate forms of genes, accounted for heritable differences among individuals. James Watson and Francis Crick elucidated the structure of DNA, a macromolecule composed of two complementary chains of nucleotides. Researchers are now able to construct and scan enormous databases containing DNA sequences to address questions about genetics. The breathtaking sweep of this approach is far cry from the simple approach that Gregor Mundell pursued in his monastery garden. Genes can now be studied at a molecular level with relative ease, and vast numbers of genes can be studied simultaneously. This approach to genetics, rooted in the analysis of the DNA sequence that make up a genome, is called genomics. It has been made possible by advances in DNA sequencing technology, robotics, and computer science. Efforts are now focused on studying how they influence the myriad characteristic of human beings. The massive DNA sequencing projects that came on stream at the end of the 20th century have transformed genetics in a fundamental way. The articles reported that 2.7 billion nucleotide pairs of human DNA had been sequenced. Computer analysis of this DNA suggested that the human genome contains between 30,000 and 40,000 genes. More recent analysis have revised the human gene number downward to between 20,000 and 25,000. These genes have been cataloged by location, structure, and potential function. However, a privately funded project initiated by Greg Venter, scientists and entrepreneurs, soon developed alongside the publicly-funded project. In 2001, all these efforts culminated in the publication of 2 lengthy articles about the human genome. The paragon of all the sequencing programs is the Human Genome Project, a worldwide effort to determine the sequence of approximately 3 billion nucleotide pairs in human DNA. As initially conceived, the Human Genome Project was to involve collaboration among researchers in many different countries, and much of the work was to be funded by their governments. We refer to the collection of DNA molecules that is characteristic of an organism as its genome. Sequencing the genome is therefore tantamount to sequencing all the organism's genes, and more, for we now know that some of the DNA does not compromise genes. The function of the non-genetic DNA is not always clear, however it is present in many genomes and sometimes it is abundant. And near the end of the century, their dreams became reality as projects to determine DNA-based sequences in several organisms, including human beings, took shape, obtaining the sequence of basis in an organism's DNA. That is, sequencing the DNA should in principle provide the information needed to analyze all that organism's genes. And if geneticists in the first half of the century dreamed about identifying the stuff that genes are made of, geneticists in the second half of the century dreamed about ways of determining the sequence of basis in DNA molecules. RNA, like DNA, consists of nucleotides linked one to another in a chain. However, unlike DNA, RNA molecules are usually single-stranded. The genes of most organisms are composed of DNA, although in some viruses they are made of RNA. Some contain hundreds of millions of nucleotide pairs, and their end-to-end length exceeds 10 centimeters. Were it not for their extraordinary thinness, about 1/1,000,000th of a centimeter, we would be able to see them with the unaided eye. In this sense, then the 2 chains of DNA molecules are complementary. A double-stranded DNA molecule is often called a duplex. Watson and Crick discovered that the 2 strands of DNA duplex are wound round each other in a helical configuration. These helical molecules can be extraordinarily large, some containing hundreds of millions of nucleotide pairs, and their end-to-end length exceeds 10 centimeters. During the research, Watson and Crick discovered that DNA molecules consist of 2 chains of nucleotides. These chains are held together by weak chemical attractions between particular pairs of bases, pairs with T and G pairs with C. Because of these base pairing rules, the sequence of the one nucleotide chain in a double-stranded DNA molecule can be predicted from that of the other. The nitrogen-containing bases are not involved in these interactions, thus a chain of nucleotides consists of a phosphate sugar backbone to which bases are attached, one base to each sugar in the backbone, from one end of the chain to another. The bases form a linear sequence characteristic of that particular chain. This sequence of bases is what distinguishes one gene from another. The linkages are formed by chemical interactions between the phosphate of one nucleotide and the sugar of another nucleotide. And the big breakthrough in the study of nucleic acids came in 1953 when James Watson and Francis Crick deduced how nucleotides are organized within DNA. Watson and Crick proposed that the nucleotides are linked one to another in a chain. In RNA, the 4 kinds of bases are adenine, guanine, cytosine, and uracil. In DNA, they are A, G, C, and thymine. Thus, in both DNA and RNA, there are 4 kinds of nucleotides, and these 3 of them are shared by both types of nucleic acid molecules. In ribonucleic acid, or RNA, the constituent sugar is ribose. In deoxyribonucleic acid, or DNA, is deoxyribose. Within RNA, or DNA, one nucleotide is distinguished from another by its nitrogen-containing base. Nucleic acids are made up of elementary building blocks called nucleotides. Each nucleotide has 3 components. 1 a sugar molecule, 2 a phosphate molecule, which has acidic chemical properties, and 3 a nitrogen-containing molecule, which has slightly basic chemical properties. The big discovery of Mendel's paper launched a plethora of studies on inheritance in plants, animals, and microorganisms. The big question to everyone's mind was, what is a gene? In the middle of the 20th century, this question was finally answered. Genes were shown to consist of substances called nucleic acids. These discoveries were published in 1866 in the Proceedings of the Natural History Society in Brünn, the journal of the scientific society in the city where Mendel lived and worked. The article was not much noticed, and Mendel went on to do other things. In 1900, 16 years after he died, the paper finally came to light, and the science of genetics was born. We shall delve into Mendel's research and its application to the study of inheritance, including heredity in human beings. The coexistence of allies in the plant, therefore, does not compromise their integrity. Mendel also found that allies of different genes are inherited independently of each other. Mendel emphasized that the hereditary factors, that is, the genes, are discrete entities. Different allies of the gene can be brought together in the same plant through hybridization and can be then separated from each other during the production of gametes. And Mendel proposed that pea plants carry 2 copies of each gene. These copies may be the same or different. During reproduction, one of the copies is randomly incorporated into each sex cell or gamete. The female gamete eggs unite with the male gamete sperm or fertilization to produce single cells called zygotes, which then develop into the new plants. The reduction in gene copies from 2 to 1 during gamete formation and the subsequent restoration of 2 copies during fertilization underlie the rules of inheritance that Mendel discovered. And Mendel studied several genes in the garden pea. Each of the genes was associated with a different trait. For example, plant height or flower color or seed texture. He discovered that these genes exist in different forms, which we now call allies. One form of the gene for height, for example, allowed pea plants to grow more than 2 meters tall. Another form limited their growth to about half a meter. And Mendel's careful analysis enabled him to discern patterns, which led him to postulate the existence of hereditary factors responsible for the traits he studied. We now call these factor genes. And good day, it's Dr. Will, CBMD, PhD. Today's Sunday, January 21st, 2018. It is 1:49 p.m. Eastern Standard Time. Mendel. Genes and the rules of inheritance. Although genetics developed during the 20th century, its origin is rooted in the work of Gregor Mendel, an Australian monk who lived in the 19th century. Mendel carried out his path-breaking research in relative obscurity. He studied inheritance of different traits in peas, which he grew in the monastery garden. His method involved hybridizing plants that showed different traits. For example, short plants were hybridized with tall plants to see how the traits were inherited by the offspring. And this is fantastic achievement by working with the very earliest cells. This study has created stem cell lines that can form both embryonic and all other extra embryonic cells. The methods and insights from this study in mice could be used to help establish cultures of similar stem cells from other mammalian species, including those where the ES or IPS cells lines are available. Yet, the research also has great implications for human regeneration medicine, as stem cells with improved development potential open up new opportunities. Further research in the area is vital so that we can properly explore the potential of these cells. And